Hello, this is my quick guide to using ImageJ. ImageJ was developed by the NIH to analyze biological images, so it's a bit more appropriate than Photoshop for analyzing things that you'll find under a microscope. So, to start off, what I'm going to do, I've got permission from my PI to use a couple of images that we've taken from our microscopes, and I'm going to show you how to use them. So, the first thing that I'm going to show is I have this stack of TIFF files over here. Now, if you're taking a time lapse or a Z-stack, your microscope may give it to you in a stack file, which is a bunch of images into one TIFF file, but this particular microscope gave it out into a bunch. Every single image is its own file. So what you do in that case is you take the entire file, put it into a folder, and drag it onto the ImageJ window. And you don't want to convert it to RGB, you want to convert it to a virtual stack. So now you have it in an entire stack over here. You can move the slider to move forward and backwards in time, and if you have a mouse on a scroll wheel, you can move that scroll wheel back and forth in order to get to a different part in the stack. Now I want you to take notice in the main ImageJ window, right where the text is over here. When you mouse over any certain point on the window, it'll give you coordinates, and it seems that right now these coordinates are in inches, but you can change whatever unit. Your microscope might save the TIFF file in micrometers, which makes everything a lot more convenient, but the top left corner has the coordinates of 0, 0 in the XY direction, and the Z value just refers to where you are in the frame on the Z stack. And the value that it shows you is going to be the grayness value, if that makes sense. So 0 is black and 255 is white, and everything between that tells you a different value that you might find. And a tool that you can use to analyze that is this line profile tool. If you don't already see it, then click these arrows over here and select whichever ones you want. And I don't really use it very much, but you might find a use for it. It basically gives you a profile along these lines. So you can tell that everything in the background is pretty light while the cells are pretty dark. So let's see what happens when we mouse over a cell. And yes, that's exactly what you'd expect. Although that's upside down, I'll do it the other direction. And it shows you high values over here, low values over here, high values again, and it shows you the line profile across that line that you just drew. Another thing you want to pay attention to is RAM, because when you have 129 images in one stack, it can use up your memory pretty easily. Right now this is running at 166.2 MB, but I've before used files that could be as many as 1,000 images long, and that can take up your RAM pretty quickly. My computer is able to handle that sort of load, so it's not that big of a deal, but make sure that you have the sort of hardware that's necessary to do this sort of analysis. It's always a good idea just to keep your task manager open so that you can see what you're doing if anything's running too slow. Another thing you want to do is, if you want to label parts on your diagram, you can use the text tool. Now, before you do anything, make sure that your image type is 8-bit. It makes all the analysis a lot simpler than if it were 1632 or RGB. That's because everything in ImageJ is done by intensity, and you saw the intensity of the black and white values. And so there's no real need to have it in color or anything very much above what you see in 8-bit, because when you have those sorts of colors, it just makes the analysis more difficult. And I'm sure that this could vary by the sort of file that you're analyzing, but in my experience it's been simpler to do it in 8-bit. Now, when you right on top of here, you can double click the tile in order to get up the context menu, and you can change the color to whatever you want. I keep it in red just because that's default and it's easiest to read. You can move around the text size, you can change the text angle, and you can write whatever you want. And if you push Control B, or on a Mac, I'm sure that's Command B, it'll stick in place. And the reason why you can have this red annotation on top of this 8-bit image, even though 8-bit technically shouldn't support color, is because this isn't actually putting it on top of the image. It's not changing the underlying image information. All it's doing is overlaying its own personal reminder on top of it, if that makes sense. And for contrast, I'll get you this paintbrush tool over here, which is set to the color red, and doing the exact same thing but actually modifying the image that we have. The computer interprets it as having the same intensity as what we think of as red. The only difference is the annotation we overlaid isn't actually part of the image, so it doesn't change any of the image data. So the next file that I'm going to show you is a Minutewise time-lapse of these yeast cells. You don't have to drag and drop them onto ImageJ, you can just click them, but I find it's easier to just do it this way. So here we'll get into how we do measurements. So before you do any measurements, you'll go to the Analyze window and set measurements, because every time you measure something, the measurement window will give you something else. So I set it to measure area, and we can forget about this for a little while. All we really care about is area. So the most basic kind of measurement that you can do is you take a box and you push Control M or Command M on a Mac, and it gives you the area of that box in whichever unit that your file is saved in. Over here you can see that it's saved in pixels. And it gives you a mini spreadsheet manager for whichever measurements you've just taken. And if you were to take the length of every individual cell, you draw a line on them, push Control M, and it gives you the length over here. It also gives you the angle that your line is drawn at, but that's not extremely useful for this sort of data. But what we want to do for this sort of file is track how these cells grow over time. So if you watch them grow, they change very slowly, and it doesn't make very much sense to draw one line and then measure that, and then move to the next frame and then draw another line and measure that. It would just take way too long. So another thing that we can do is use the point tool. And you double click on the point tool, and what you can select is auto measure and auto next slice. You can choose to do add to ROI Manager, but that's not what I typically do. I'll get into ROIs in a little bit. And you push OK. Now what this does is it allows you to, instead of having to draw a line every single time, what you can do is focus on just one endpoint of the cell and keep clicking on it, and it'll automatically advance to the next slide. And that way you can get these measurements done a lot quicker than if you'd had to draw a line every single time. And then once you're done doing that, you simply go back to the first frame and repeat the process again with the next endpoint. And obviously you'd probably dump that into Excel before you start over. And by doing that, you can put in a little formula to extrapolate the area of the line that you would have drawn, and that makes your measurement A, a lot quicker, and B, a lot more accurate. And I already showed you the color profile tool, but I'll just repeat it again. Over here we have what we consider a dark background, and if you see the little halo around each cell, that'll give you an idea of exactly how ImageJ thinks of these images as intensities, color intensities. And that'll get important in a little bit, because we're going to do something called image calculator, which I'll add to my to-do list. I believe that that would be somewhere over here. So this image over here, visit 2, is probably going to be where you learn most of the useful stuff over here. How this image file was saved is as a dual vision file, which basically stitches together the GFP and the phase images. Now what we do right now is, since we only want to analyze a couple of the images at a time, we can take this stack and break it down into its components. So there are a couple ways we can do this. So we can go to Image, Stacks, and we can say stack to image, and that takes the entire thing and decomposes it into its constituents. But this thing is 12 frames long. It's not, when it's 12 frames long, it's not that big of a deal how big it is. But let's say we had something 300 frames long and we just wanted some frames in the middle. So in that case, what makes more sense is image stacks, and we want to make a substack. And give me a minute to find exactly where that is. Huh. Could have sworn it was there a minute ago. Make substack over here. So I only want frames 1 and 2, so I'll type in 1, 2 or 1-2, whichever you want, and that outputs a substack with just the first 2 frames, and they don't necessarily have to be continuous if you use commas. And now that you only have the frames you want, then you can decompose it from stacks to images. So now here I have my phase image on the left and my GFP on the right. And the GFP is just of the nucleus. So what we're going to do with this image is quickly save it, because we're going to be modifying it and then doing more work on it. So we'll call this the GFP. Now we can take this GFP image and compute its threshold. This is an extremely useful tool. Before we do anything, just make sure that it's in 8-bit. It was 16-bit before, but if we make it in 8-bit, it makes the threshold analysis a lot more simple. So image adjust threshold, and I'm taking you through all the context menus just because you should get a feel for it, but whenever I do it, I just use the keyboard shortcuts. So you drag along this over here until you get what you want, and I want to just select these nuclei. And if you have a dark background, select dark background. If you have a light background, uncheck it, and it'll basically just invert whatever you have. And it looks like I've selected what I want. So I click apply, and now what we have is a binary. A binary is just black and white, and it's not useful for actually computing things. What it is useful for, though, is getting regions. Now if you look closely over here, it's a bit rough around the corner, and if you look at this pixel over here, that's considered its own region, meaning that it's not continuous. So we can fill in these holes by going to process, binary, and fill holes, and suddenly all those little obstructions to what we wanted are all gone. So now we have these. Go to analyze particles, and this is probably one of the most useful things. So when these boxes over here, you can choose to change them. Right now it doesn't really look like there's very much to change, but size in micrometers, that basically says, do we want to analyze all particles that you see over here within a certain range? So all of these look pretty big, but say if you had a couple of white specks over here, maybe we'd say, yeah, we want everything to be greater than 1 micrometer before we operate on it. It also goes for circularity. If you see this oblong piece over here, we want to change the oblongness of it, and I'm not exactly sure. I think 1 goes to perfectly circular and 0 goes to not circular at all, but don't take my word for that right now. I don't use it very often. More often what you'll want to do is just set your lower bound to about 1.5 microns, and if you're not completely sure how big that is, what you can do is just draw a little circle of your own and measure that, and yeah, that's about 1.5 microns, and everything bigger than that will be selected. But since everything we see right now is already pretty big, we can just go ahead and make sure that you have add to manager and exclude on edges. What exclude on edges means is it won't count this particle over here because we only see part of it, and that's not very useful to us. So add to manager, and now we have ROIs, regions of interest. So before I do anything on the ROIs, I'm going to go through them all. I'm not sure if you can see very well, but they're now highlighted in yellow. And make sure that every ROI there actually corresponds to an entire nucleus. So I'm going to go through and delete everything that's not a nucleus. Actually, that might take too long. So you get the idea of what I was trying to do. Now that I have that, I can close the modified GFP image and open again the original GFP that I had, because now we want to actually do analysis on it. The ROI Manager simply keeps your regions, your areas of selection that you already had, and now we can actually operate on it. This is where we're able to get useful information. So now we select all of the ROIs and we set the measurements, what we want to measure from it. We can take the area and we can take the mean gray value, and I'll open up Excel for us to dump these values into. The annoying thing about ImageJ is that everything is in a separate window, so every time you close it you're going to have to open it up again. So select all your ROIs at the same time and click Measure. Actually, before we even do that, make sure our measurement box is empty. Yep, it's closed, so that means it's empty. And click Measure. And now for each ROI you have over here, it gives you a corresponding area and mean value. So we can paste this into Excel, ROI, area and mean gray value. Now the next thing that I did for this project was work on the phase images over here, where I would go to the first ROI and find out which cell that corresponds to. And yes, this is a bit annoying that ImageJ closes each one of your windows every time you're done using it, but it's eventually something that you just grow used to. So I had measured the length of this first cell over here and then measure this next cell over here, because we were trying to find a correlation between the nuclear GFP intensity and the length of the cell. But that's not very important right now. Let's just assume that I have another column over here called length, and I put in their length values. And then over here we'll have, say, total GFP expression, and that'll be equal to the area times the mean gray value. And then you just drag that all along down here, and you've got your mean gray values. So that is just an example of something that you could do. Let me look through my to-do list to see what else we can do. We've covered stacked images, substacks, image thresholds. We've filled holes, created ROIs, measured. Merge channels, that's another interesting thing that you can do. So because ImageJ considers everything as intensities and you don't really need to worry as much about the colors themselves, we can create composite color images a lot more simply than you could in, say, Photoshop. So I'll open this up again. Oh, I already have this open, so I'll just open the GFP slide. And let's say we wanted to make the GFP appear just in green and then the phase image appear in red. You do image, stack to RGB, I believe. No, merge channels. That's it. And the one on the left, we'll look at their title bars. This is called Substack 001, so we'll make that red. And this one is GFP, and we'll make that green. And it'll create a composite. Oh, they have to have the same bit depth, so we'll make sure this is 8-bit and this is 8-bit. And now we'll do that again. Image color, merge channels, And look at that, you've now got a good looking GFP image that you can put in a paper. And now the last thing that I'm going to get to is this, still 001. And there are a couple of things that we can cover over here. I didn't cover donuts in the previous one, so I'm going to move that bullet over here. So over here we have a phase image and a very overly exposed GFP image, and this looks extremely washed out. What are you going to do about it? Well, you'll go to Image, Adjust, Color Balance. No, not Color Balance, there is no color here. Image, Adjust, Brightness, Contrast, and Auto, and look, you've magically gotten a good looking GFP image. And for this GFP, it also gives you a bit of the outline of the cell because it's also there in the cytoplasm. But look what happens when you go back to the first image. Oh no, it's completely dark. Let's see what happens when we auto brightness this. Oh look, it looks even better than before, but the problem is now your GFP images look washed out. So what we do in that case is, again, we decompose it, stack to images, and now we can operate on them individually without them altering each other. So automatic on this, automatic on that. And I forgot what I was going to say, but I'll say it whenever I remember. So over here, if you'll notice, this is a GFP image, but it doesn't look very much like what we had before. Part of the reason for that is because there's a background. In the previous GFP images, everything in the background was black. So what we can do is subtract the background in order to make it like we saw it before. So process, subtract background, and it uses what we call a rolling ball algorithm. And you can think of it like this. So because everything that ImageJ sees in every pixel is a power between 0 and 255, you can represent that with a three-dimensional graph where the base is the x and the y of the image, and the height is the power of the intensity value. And you have a rolling ball that's rolling around there, and it's physically rolling around on this 3D graph. And if the radius of this ball is 50 pixels, it'll subtract everything that it's landing on. You kind of have to have some sort of visualization for this to completely make sense, but I'll click Preview over here, and it's erased everything in the background that was within its 50 pixel radius. Let's see what happens when I increase that to 100 pixel radius, and now it's erasing slightly less, and 500 pixel, and it's erasing even less and less. So you can kind of imagine that there is a ball rolling around on this image trying to get into the nooks and crannies to clear out the background. Let's see what happens when you do 5 pixel. It's even clearer. And that's what we do for erasing the background. Back to my to-do list. Auto balance, rolling ball, donuts, and image calculator. So I happen to notice, I'm not sure if this is correct, but I think you mentioned something about how you wanted to measure the average intensity a certain distance from a cell. And I've already shown you how to use ROIs to measure the average intensity of a cell itself. But how do we measure from a certain distance? Well, we'll go back to our adjust threshold, but before we do that, make sure it's an 8-bit. Image adjust threshold. This is a bit less clean. Although it doesn't completely cover everything we want, let's say this is good enough. Apply. Alright. And we'll keep this over here and save it as a TIFF, and we'll call this binary.tiff. And before we operate on this, we'll close up any of the holes that I just zoomed in on. By the way, I zoom in by pushing the up and down buttons on my keyboard or by plus and minus. Process, binary, fill holes, and the holes are gone. And this is what I was talking about before, how these little things that don't have very much area, you can ignore them when you're measuring your particles. Now here's a tool that you can use, process, binary, erode, and dilate. Erode and dilate takes every single one of these individual regions and it either makes them bigger or smaller by, I believe, about 1 pixel. So I'll go to this very sparse looking region over here, for example, process, binary, dilate, and it makes it bigger. Process, binary, dilate, and it makes them even bigger and bigger. So let's say that I want to keep on dilating them, and erode does the exact opposite if you ever try it out. Dilate, where'd it go? Process, binary, dilate. Okay, and let's say I decide that's good enough. Now I'll open another copy of this binary.tiff where all the regions are smaller. And here's where ImageJ's magic actually comes in. Remember how I said that ImageJ thinks about images as numbers? Well, now we can use what's called the image calculator. And we use the image calculator. Image calculator. Oh, I should really name them different things. So I'll save this as binary expanded. Okay. So process image calculator, and it does essentially think of it as a matrix of power values where every single pixel is its own element. And now we have binary expanded, and we want to subtract binary 1. And we'll create it in a new window. So theoretically what this should do is take these white regions over here and subtract these white regions over here, because remember, white is 255 and black is 0. And if these are the same bit depth, yup, you've created a donut. And that's, I think, what you were looking for if you want to have cells and then measure from a certain distance away from them. You've now created these white regions that you can now add to your ROI Manager. So measure, analyze particles, and I'm not going to bother with the settings in particular, but now look, you've gotten a bunch of selection regions where everything's in a donut shape, and if you were to open up your original image over here, you can now measure those donut-like regions on the original image. Wait, I don't know exactly what I did, but well, you get the idea. The donut shapes are still present, and now you can use them to measure. Another thing that I just remembered that I can bring back is it's mainly a cool little thing that you can do on binary images. So adjust threshold when it's in 8-bit, adjust threshold, and make that binary, black background, yes. Okay. Now let's focus on just these two cells over here, right? Now imagine that these weren't separate. Imagine they were a bit closer together, and I can simulate that by process binary dilate and keep doing that. I find it a bit annoying that there's no quick shortcut for that. And now they're actually touching. And if ImageJ were to analyze this, it would think of it as just one continuous region. So what we can do in that case is process binary, and this is called the Watershed Algorithm. And again, think of a 3D visualization, and where 255 are the highs and zeros are the lows, and it would essentially throw water, pretend you're pouring water onto these regions, and it would automatically segment them like this. And you can Wikipedia it for a better visualization, but I'm going to click on it, and it essentially just does this, where it segments things that it detects are close together but aren't the same thing. And this is useful for if you're trying to keep track of individual cells, especially with a resolution that makes it hard to get a good focus on it. And there are also a couple of other algorithms that you can play around with. If the video suddenly cut off from before, that's because I had made a mistake, so I'm just going to re-record it. Another cool algorithm you can do in binary is Verona. So you start with a binary, and let's just say that you have dots over here, white dots, which in your actual binary will be the outlines of cells or nuclei or whatever you want. And the Verona algorithm basically demarcates boundaries where boundaries that are closest to each cell. And that might not make very much sense right now, but I'll show you what I mean in a minute. I'm not actually going to save that, I'm just going to duplicate it over here. And so process binary Verona, and when you overlay this back onto the original image, image calculator, you add these two, untitled 1 to untitled. It basically divides everything into regions where everything in each region is closer to this dot than to all the other dots. And that might not look like it's very applicable to what you need, but actually if you want to segment things in a way that the Watershed algorithm doesn't cover, this is the best way in order to differentiate two different cells or nuclei from each other. And I think that about wraps up what I wanted to discuss in this video. And the only other thing I can say is you just got to play around with all the different features in ImageJ before you really get a feel of it. There are a couple of other things that I haven't really gotten very much experience with. That would be putting in plugins. You usually just download them from online. And another thing would be writing your own scripts, macros that you automate. So macro, you can record a macro. Every single thing that I've done can be automated just by recording a macro, and then if you do something repetitively, you can just do it to every single image that you need without having to manually do it every time. The only downside to that is sometimes sensitive values will change depending on the sort of image you want, but overall, if all of your images are consistent and you'd have to repeat that multiple times in a row, it's pretty convenient to automate. And again, you want to make sure that you have enough memory in order to operate this all, and check your hardware limitations before you put on ImageJ, and just for a good measure, make sure that not too many other programs are open while you do this, and of course save your work often. That wraps up my quick guide to ImageJ, and if you have any more questions, just look at the documentation, or if I'm available, email me.